All right, we're recording now. So we're going to get started for today's session, where we're going to be looking at how to process single-cell RNA sequencing data that we get out of the cell ranger count function into a fully processed Seurat object that can be well utilized for a bunch of downstream analyses to make a lot of pretty plots. So first, we want to acknowledge that we're on the traditional, ancestral, and unceded territory of the Musqueam people. This is where we're presenting from today at UBC. Here's our copyright information slide. And here are the learning outcomes for this session. So first, we're going to use cell ranger, make, FASTQ, and count outputs to create a Seurat object of our single-cell RNA sequencing data. So earlier in the first session, we already used the cell ranger, make, FASTQ, and count functions. And so now we're going to see what we can do with these with Seurat. Second main takeaway, main learning objective is using two packages. And now there are many packages you can use, but the two packages we're going to employ are SoupX and Doublet Finder to help process the single-cell RNA sequencing data just a little bit more, knowing the biology of how the single-cell RNA data set was made with this droplet-based 10x kit. Lastly, we're going to perform ST transform on multiple samples. On multiple sample objects that we make at once and then integrate those objects so they're all put together for downstream analyses and comparisons. Here's our afternoon workflow. We're going to start by launching RStudio on Sockeye, go in and use that SoupX function or SoupX package to remove background noise and ambient RNA. Next, we're going to bring the outputs of that SoupX function into Seurat, and we're going to do some labeling. We're going to make some metadata labels. After that, we're going to do some quality control and thresholding, and there'll be a bit of a dialogue there, discussion there about what parameters set, why, things like that. After that, we're going to remove artificial doublets from the data using Doublet Finder. Lastly, we'll scale, normalize, and cluster using ST transform on multiple data sets, and then we're going to integrate those multiple data sets so they can be used all together for differential gene expression, trajectory analysis, whatever you want to look at for your single-cell RNA sequencing data. Awesome. Okay, so moving forward, I have this giant disclaimer here because I was worried that this would be a long session. It's not. So we initially had like this reverse coding approach that you might have picked up on with what we did this morning, where we would basically submit our pipelines. And as they ran, then we opened it up to look at the script itself and dove into the script essentially. For this, it's going to be a little bit faster. So it won't be as much of that reverse coding approach. We'll just kind of talk through things as we go through all the steps. So please ignore the second bullet. Like there's a ton of information, but we do have the time for you guys to stop and ask questions as we go through. There are many different ways as well to process single-cell RNA sequencing data. There's a lot of publications out there as well, comparing the many different ways to process single-cell RNA sequencing data and how to process it. And here's another package, so you can process it this way. And oh, here's our spin-off of that package. So there's a million ways. I'm showing you just one. So consider your data, consider what works for you, consider the pros and cons of the packages, because there's a lot of input. You're going to know your cells and your biology the best, see what works. This is just one way. It's not the best way. For me, it's the best way for me, but there's a bunch of other ways. So this is an application-based workshop. We're going to be looking at how to use these packages, these functions, and apply them to our data set. So I'm not going into as much though of like the math behind everything. I will refer to the publications about these packages if it starts getting close to the math, because the publications are going to do it a lot more justice than me pretending that I completely understand what's going on and can regurgitate it. So let's get started. We're going to launch RStudio on Sockeye to begin. So everyone should be logged into Sockeye right now. And if you're not, hop on. Knowing how to do this as well is technically a prerequisite for this workshop, but we are going to make sure, since we do have the time today, we're going to make sure everyone gets this up and running because this is how you're going to be able to see everything. So step one, you're going to go to your single cell RNA sequencing directory. So if you've been in any of the other workshops last week anyways, you would have this directory already made. If you don't have this made, Alejandro can tell you how to make this directory in the chat on Zoom. But can everyone just give me a quick thumbs up that they have this directory while they're launching this? Awesome, awesome, awesome. Okay, I'm seeing hands up and thumbs up in person. Good, thank you, Zoom. Great. Okay, so if you don't have this directory made, I'm just gonna check the chat. Alejandro is gonna paste the command. Thank you, Alejandro. So you're going to submit run RStudio.sh. You're going to basically open up this connection file and follow the steps there. After that, you're going to open a brand new terminal or whatever you're using for this. And you're going to put in this, but yours will be different and unique to what it says up here in this connection file. So follow the steps there. Copy the part that looks like this, but tailored to you with SSH into a new terminal, and then follow those prompts. And then point your browser to this address here. I'm going to give you guys a minute to do that. People in person, how are we doing? Good, you're there. You know, you don't have access. Okay. All right, Zoom, can you give me a thumbs up once you get RStudio launched?
Did we mention that try to copy the link paths on the slides?
Yeah, I actually will talk about that. Yeah, once they get in there. So it's because see these we'll get to that. We'll get to that. I'm too far ahead. I can't actually go back. Oh, there we go. Okay, everyone has RStudio opened on Zoom. Thumbs down if you don't have RStudio opened on Zoom. And please reach out to Alejandro if you don't. No one's giving a thumbs down, so I'm going to assume RStudio is open. All right, and it is critical that's on Sockeye, just so you guys know. So once you have RStudio open, I want you to change. So type in lib paths and then this file path, okay? And the reason why we're typing it is because these little quote marks here, R does not like them. They like the ones that are basically just a vertical quote mark. So when you're typing it in, it'll automatically adjust that. That's why you have to type it in. So you need to set your lib path, so your library, to this library that I've made here, which contains the critical packages we need to run what we're running today. Okay. And to check that, you just type in .libpaths and then hit enter, and then you should see this. So this should be in your console. Good. And then feel free to configure RStudio how it best suits you. This is how I do mine. So people might feel a little flipped around. If you're dozing off for now and you come back, or pay attention, basically, my console is on the top, my source is on the bottom. So if it's easier for you to do it that way to follow along, do it. If you're more comfortable having it with the source on top, console on the bottom, feel free to keep it that way. Just know that mine is not, mine's flipped. All right. Okay, can everyone give me a thumbs up that you have the library paths properly referred to? Yeah, thumbs up, thumbs up. In person too, thumbs up if you have it. If not, we can help. Good. Okay, Zoom, I see one thumbs up. Okay. Anyone thumbs down? Can anyone not do it? I do want to know before we move forward. No thumbs down on Zoom? Okay. If you do need help, reach out to Alejandro. He's our TA on Zoom today, but we will keep moving forward. So just going to move you guys off to the side, get rid of the chat. Things look good. Okay, so first things first, we're going to remove background noise or ambient mRNA using SoupX. So SoupX is a package that does just that. And the way it does this is it compares the raw featured BC matrices that we get from our cell ranger count output to our filtered ones. So it's basically comparing what the cell ranger count function started with, so those raw reads, and then looking at what it ended with, the filtered ones, to get a sense of ambient mRNA in the background. This is going to happen no matter what droplet-based library preparation kit you're using. It's always going to happen. It's just a matter of, are you getting rid of all of it or not? And for very clean data sets that are in the chip loading stage, it's very cleanly loaded. This package will really not remove anything that isn't necessary. Otherwise, if you have a single cell RNA sequencing data set like I've had before, where during COVID, there were certain stop points, the cells were kind of sitting in limbo, the cells got more stressed, they start leaking mRNA, and there was a ton of ambient mRNA in the background that we actually had to go back through and filter through that cell ranger count missed. And that ended up really tidying up our data set. So that's what this function does. This is what this package does. So I'm going to switch over now to RStudio, which has disconnected. Okay, we're just going to reconnect quickly. All right, so we're reconnected to RStudio. And the first thing we're going to do is in your directory here, you should see SC process, and then you should see the other folders that you were familiar with from this morning. So data and cell ranger. So first, let's just pop into our cell ranger folders and look at our outputs. Or sorry, we're going to pop into our data folder and look at our cell ranger outputs. So data, and now we're going to go to PBMC so our C data set. Again, this is the data that was the cell ranger, the chip, the 10x chip that was loaded onto our Chromium controller machine, which is a device. This machine basically takes in this microfluidic chip and partitions the cells, the gel beads, and the partitioning oil into those gems. So a bubble basically containing the gel beads with unique molecular identifiers to label the cell as well as the cell itself. So this is using the controller machine. Let's look at our outs from this morning. This is from our cell ranger outs. If you did not run cell ranger out from this morning, cell ranger count, you're going to go to PBMCC and then into our output files folder. So PBMCC outs, you'll go into your outs folder down here, and then these are the outputs that we got from cell ranger count. The ones that we're going to be taking in and reading in for this first part is going to be your raw featured BC matrix, as well as your filtered featured BC matrix. So these are the file paths we're going to refer to with this package. And I'll just show you quickly with the output files. They're named a little different, but same thing. You want the ones that end in filtered featured BC matrix, as well as raw featured BC matrix. Okay, the chat's blowing up, so I'm quickly going to peek at that. Okay, yeah, and if you're having any errors with the .lib paths, looks like it's getting addressed. Okay, it looks like everything's getting addressed. Awesome, okay. So we're going to go now from our data folder. Let's go back to our home folder. And we're going to go down to SC process. And we're going to open up script number 1, which you can already see here. Number 1, SoupX template. So we're going to make this bigger so everyone can see, so I can see. Okay, so first things first, check your libraries. So we're going to do that. Okay, so I actually also have to set mine up really quick. So I'm just going to pop over here. And then I have to type this all out as well, if it lets me. Okay, I'm just going to pause my share, leave this up, and do this quickly. Great. Okay, I'm back. And I'm also going to test this with you, make sure that's not the right one. Okay, I'm going to pause share one more time and get mine going as well. Oh yes, I can increase the size. I will definitely do that because I actually, yeah, because I think on my screen too, it should be a little bit better to be bigger screen. Perfect. There we go. Okay. Okay. Okay, so you should see me on RStudio now. It's going to increase the size of everything. Is that too much? It seems to take up a lot of space. That's good. Okay, in person, we're good. Online, what do you guys think? Seems okay. Okay, we've increased it a bit. I'll watch the chat in case I need to increase it a little bit more. For now, I'm just going to bring my files just over to the side because we're going to be using everything within our RStudio notebook here, which is in our Markdown. So first things first, we want to bring in the libraries that we're going to be using with this script. And I have this in an R notebook format, mostly because we're going to go step by step through and we're going to be able to generate and view the outputs as we go, which I want you guys to be able to do. So press play and run and install these, bring in these libraries. So we're going to get a lot of notes here for what's going on. It's going to tell us play by play what's happening. I'm just going to exit out of that. The next thing we're going to do is we want to put in the name of the folder that this sample is coming from. So this is coming from PBMCs. So this is our Chromium data set. Next, we're going to create a directory. So our sample folder name is based off of what we have.
Asks for it. Like,
oh, I don't think you guys do. Okay, that's okay.
Oh, okay.
So type that in and add the name of the folder that will be in. So put this in because this is going to feed directly in to here, to names down here. Okay, I'll give you guys a moment to do that. I think that's the only thing I tweaked from probably an hour ago. Oh, yes. So if you guys are curious and haven't used R Markdown before, to insert a code chunk, it actually tells you up here. So you can do Control Shift Enter or Command Shift Enter to insert a code chunk. So if you're on a Mac, it's Command, I think it's Command, oh, that's to run it, sorry, Command Option I. Oh yeah. So to insert a code chunk, you do Command Option I, or you can just write it within a code chunk that's already there. Let me get rid of that one. So again, yeah, if you guys are new to our Markdowns or our notebooks in RStudio, A code chunk is everything that's within these first 3 little tick marks, and then you define the language here, and it ends within these tick marks. So every time we run something, it'll run everything that's within that chunk of code. Does that make sense for everyone so far? Are there any other questions, either in person or on the chat, that you have about this before we carry forward?
Sorry, how do you create a new code chunk?
So to create a new code chunk, I know on a Mac it is Command Option I. So for you, it might be Control Alt I, potentially.
Yep, that's it.
Control Alt I. Okay, so if you're not on a Mac, it's Control Alt I. For today, though, and for these scripts, all the code chunks are already written for you. So we're going to be just stepping through this to begin with. So the next thing we're going to want to do is create a folder called SoupX. So I'm actually over here. I'm going to go pop into my PBMCC's C folder and just see that get created. So dir create are here, and then we have the name. So it's going to basically go start in our home directory, select our SC process folder. So home SC process folder that already exists. It's going to then append what we defined as name, which is up here. So PBMCC is our name. So it's going to jump into that PBMCC data set this way, and then make a new folder called SoupX. Okay. Next thing we're going to do is we're going to use this as a file path. We're going to name that file path SoupX path. So now, in order to get to this folder that we just made, we have this that we can have inserted then down the line in the code that we can refer back to. Okay. And then I also want to add in a path to our data folder, and that's just data path. So to run things, you can hit Command Enter or Control Enter, or you can run the whole chunk at once. So as we were talking about earlier, if you ran cell range or count, you're going to use these file paths for your output. So earlier we went to our data, PBMCC, and then we went to our outs, and then into our outs folder again, and we found the filtered BC matrix and our raw feature matrix. If you did not run Cell Ranger, which I don't think many people did because it takes hours, you're going to change these file paths and use the output files that are already downloaded when you downloaded the data. So here we have name, we've already updated. Now I want you to run data dir filt, and then this line. So run all of this in order to basically put the file path, define the file path as data dir filt to be where your output files are.
Character 0.
For list files.
When I tried running it.
Oh, I am too. Yeah. I am as well. Let me check up here first. Okay, so that one's working. So if that's the,
I think in the output files, the folder has this, or it has a slightly different name. It had like the 500 stuff.
yes.
We just need to paste in that.
Yeah, so you'll have to paste in that prefix, I believe, for after data PBMCC's instead of data PBMC's C, it should be
500.
yes, it'll have to be the folder that your output files are in. So you'll have to change the name to where your PBMCC data is. So let me actually walk you through how to do that then as well, in case anyone else has also picked this up. So PBMC's output files. So it should just be yeah, so to the FASTQ matrix files, it shouldn't actually be that. So it should just be, I think this. There you go. So these are to the FASTQ let's see. Yeah, you need it. You have to append the name to the actual name of the feature matrix. Yeah, you're right. So I'll fix that as well. So 3P. And let's make sure that matches. So the ones you're going to put in this name up until, yeah, you're basically putting in this name and you basically want to refer to the folder containing your FASTQ files. So if that one has a name, update the name.
I tried adjusting it and I'm trying to like auto complete so I don't have to sync it out. And it's not like when I start typing 500, it's not coming up.
I don't, well, do you want to show her that? Or here, I'll copy these file paths into the chat for everyone. Perfect. Nope, still not getting it.
Yeah, that's
Jane, can you hear me?
Oh, sorry. I can hear you now. Yes.
Okay. So I think that these barcode features and matrix files, they are not on those in that location, because actually if you see there's no folder within the output files folder that contains these files. I think you have them in the output of the counts job. So one option would be for people to copy those two directories because they're not in these output files. But you have them in the output files of the counts job. I don't know if that makes sense.
Yeah, so I think it's also because, well, they should be, yeah, we can definitely copy those. So if you guys want, I can write in for how to copy those, or the other option you could do too is to unzip these files, I believe, but they should be unzipped. Okay, I can write something so you guys can follow along with that. So if you give me one moment, I'm going to just type that out for you guys.
maybe we just specify the folder that out files. Oh no, I see.
I'll just have you guys copy from my Scratch directory, and then I'll just write the code for that to make sure it gets to you guys from the right spot. Just have to think of where I am right now.
Probably easier to do it on the terminal.
Yeah. Oh yeah.
I would just do it on the actual.
That was the other thing. Yeah. I might just do echo path. No. Yeah. Okay. Okay. I like how Alejandro's solution too. You can also just untar the PBMC folder, but I'll put this in for those that want to copy as well. We're gonna do.
You're missing a UBC.
Thank you very much. Okay. Out and then. Oh, that's interesting. Okay. Let's see if you guys can just tar-xvzf, the two folders, the two compressed folders to get the actual folders that contain the FASTQ files. Oh, I'm need to resume share, yeah. I'm gonna get a little reoriented. All right. A new terminal,
You haven't resumed yet.
Okay. Where would you like me to go?
Yeah, so you can run that tar, show them where to go.
Yeah.
And run the tar.
Perfect. Okay. I'm just gonna get rid of this. Okay. So in here, I'm in my student space in SC RNA-seq. We're going to change over to our data folder. So I'm in terminal, CD data. We're going to go to PBMC's C. So CD PBMC's C. Now we're going to go to our output files folder. Okay, we're in our output files folder. We're going to look at everything we have here. And now we're going to run what Alejandro put in the chat to unzip these files.
Tar dash.
I don't have access to the Zoom chat. So I wasn't in the chat.
So tar-xvz, the two files you want to unzip and let that unzip. Okay. So now we should be able to see them when we hit ls in our folder, and then in the code down below. So we see them here. So in the code down below, now that they're unzipped, we're going to. Oh, yes. Sorry, it's tar. Here, I'll scroll up to it so you can see it. There you go. I'll leave that up there for you. Okay, so now you have your data, you have your path name, and you have the folder. And this should be just filtered feature BC matrix. I'm just going to make sure that worked. I'm just going to pop over to the console really quick, check that it did. It did. Good. And then same for this one too. You should not have the feature name in front of it. It should just be output files, raw feature BC matrix. So same as before, the only difference is you had to untar, just open the folders that you've downloaded, open and unzip those files. Okay. And then to check after you've assigned that, let's make sure that what we're referring to in these two data directories contains 3 files. It should be your barcodes, your features, and your matrix. Okay. Good, good. All right, Zoom, can I get thumbs up if you're good, thumbs down if you're not good? Good. Otherwise, things will be slower to address. Good. Okay, thank you, Kate. Thank you, Daniel. Thank you, Fang. Thank you, Irvin. Awesome. Okay, so we'll keep moving forward. Just going to bring myself back to my cell ranger over here, not there, sorry. We're going to go down to SC process, and I'm just going to pop back into my PBMC's data, and then we have our SoupX folder here. Okay, so now that we have our file path straightened out, which again, this is the main thing that you'll be tweaking when you get your own data or you're using different data, is going to be the file paths really for these scripts. So Now that the variables are assigned, which is what we're referring to, we're going to hit run on this code chunk below. So you're going to hit play. And what this is basically doing is taking in these two different matrices. So you have your filtered data and then your unfiltered data. And it's going to create a soup channel, tell you how many things there are. I usually like to comment in what the output of that is. We won't see this for now, but it's also going to create a Seurat object, run all these things on it, and then spit out some information. So here are your outputs. This is just some information showing what to set certain parameters at based on what we see here. This is, you can read more about every little step 2 of this package, the SoupX package, if you refer to the references on our GitHub. But the main thing that this function does that we want is it's going to spit out a new version of our 3 files that we took in. So we're going to go to SoupX over here and look what's in our folder. We have our plot that it generated just as quality control, and then it's created a new folder called SoupX Built. So this is our filtered G matrix, and we have our files that we're now going to input into Seurat in our next step. Okay. Did everyone make it through that? Can I get a thumbs up if you got your filtered SoupX output? Good, good. Zoom? Good, good. Okay, so we kept that very simple. Awesome, good. Okay, so typically this is a package you'd bring in. You would type this all out step by step. This way you can do it on your Sockeye server in your RStudio environment. It's all laid out for you. It's commented out as well. You can see some examples for what I said for how many features I have. But the main thing you have to input are just your filtered feature BC matrix and your raw feature BC matrix. Okay. All right. Just going to hit save on that. The next thing we're going to go to is our metadata. We're going to start labeling our object and our metadata that come in with the Seurat object. So go and open in your home directory, and then SC process, you're going to go and open your second R Markdown template. So open that up. And one more thing to mention too, all of these libraries that we're loading in, that was the critical part. Every time you open RStudio or launch it, you're going to want to refer to the file path that we referred to for our libraries at the very start, because these libraries live in that container. Otherwise, you'll have to download them all yourself for your own spot.
So if we want to use our offset of out of Sockeye, we have to download all of them.
Yeah, so if you're not on Sockeye, you'll need to download all of these. So if you're on a different server.
Once they're downloaded into their folders, you just have to open them in R?
That, yes, yeah, so you open the library by these functions here. So you go library, open. Those are questions to probably, I would say, go through our RStudio and package management workshop series that was recorded and put forth. That would be a good one to refer to for this sort of stuff. Yeah.
I think that one, I went to that one. I think that was on Sockeye too. I'm just curious about outside, but that's okay.
Outside of Sockeye, you kind of just follow the same steps for installation. Some of them take longer than others to install, and they have a lot of dependencies. So for neat and tidy stuff, it's nice having being able to run this on the server, because we have all the packages we need and we can add to it, and then people can still refer to our single-cell RNA sequencing library container for other single-cell RNA sequencing that people do. All right, so for this next bit, we're just going to make sure these libraries are loaded again. Most of them, I believe, already are. And so for each sample we get, we'll kind of repeat this process. But we're going to start by reading in the data with our read 10x function. And that's going to read in the outputs of cell ranger count pipeline, or what we also just brought into SoupX and then it's spit out. So again, we've got the name of the sample. So this is just some file path organization. We're going to create a couple more directories within our PBMCC folder for this data. So we're going to do dir create filter outs, which is going to be for our filtered outputs. And we're going to name that path with this here. We're going to do the same thing for the next two sections. So pre-filtered outputs, this is where we're going to put some plots before we filter our data. And then the filtered ones are going to be for after we filter our data. And then lastly, I want to make a folder for our objects. So these are going to be for our Seurat objects. Okay. So we're going to define our data directory. And this is going to be, instead of where we had our cell ranger count outputs, we're going to have it refer to our SoupX filtered folder that we have down here. So in here. So now we're going to be taking in these instead of the cell ranger outputs. Let's make sure that these still exist in here by making sure we list the files in our data dir that we want to see, and we do see them, our data directory. Great. And so now we're going to use our read 10x function to take these files in and put them into data. And then finally, this is going to take a little bit of time. We're going to change that. So our counts should be data. So we're going to go create Seurat object. Our variable counts is going to be the data that we just brought in. Our project, we can title that whatever we want. For this, we're going to call it PBMCC. We're going to set the minimum cells that we want coming into our object to be 3. And so what this means is for the min cells variable, it's going to only take in cells that have like a minimum of, I think, 1 gene. If there's 1 gene, it's only going to bring in cell to make it in, or that gene to make it into the data set. And then on the other side, we have minimum number of features. So that's going to only take in cells that have at least 200 genes. And here, features is almost interchangeable with genes, by the way. So we're going to run that, and that's going to create our Seurat object. In the past, getting from here, like our cell ranger count outputs to here, was a very complicated process that has become a lot more streamlined as the Seurat package for single cell RNA sequencing has been updated and made better. So now it's become a much simpler process. And this is pretty standard to set these thresholds as such. Okay.
The min feature means min genes.
Yeah, so it's going to bring in cells that have at least 200 different genes. And then min cells will bring in genes that are in at least 3 different cells.
Okay.
Yeah.
Do you ever get a gene as a cell?
I haven't looked for one per se.
Lowly expressed things.
Well, if that's the case, then it's probably not a gene that's expressed in 1 cell. Could be an error.
No, just if it's lowly expressed,
You can't,
because you're
yeah.
sampling, you could get 1 read in 1 cell.
You could, and that's where these parameters, and this is going to be my big thing for this section, is all these parameters are, of course, tweakable. Don't just assume because I did it or a tutorial did it or a vignette did it that this is the set standard. It's really, again, going to depend on what you're expecting to see in your data, what you know about the biology and what you're looking for. So it's almost like a craft when it comes to filtering and kind of thresholding your single cell RNA sequencing data, because you do have to consider the biology a little bit. Some people do, especially bioinformaticians, statisticians, will kind of set just like, oh, we're going to get rid of the lowest 5% of these, highest 5% of these. We're just going to kind of cut off the ends of the spectrum statistically, but then you could be missing things that you want to pick up, that you want to see, that you're anticipating, you're looking for. So if that's the case and you're missing things from your data, go back, and get rid of, like, drop this down a little bit. But the reason why we have it set as 200 is if there's a cell with less than 200 features, especially with this data set, which is a well-tested classic example data set, then the odds are, not even the odds, but it's likely that it is not a full cell, that it is ambient mRNA still in the background. So that's us setting that minimum threshold, knowing that we're getting these reads in the sequencing data from droplet-based single-cell RNA sequencing. So since you know how things happened from the bench side of things, that the bench side will and should influence the bioinformatics side and what you're doing statistically as well. So it depends. Okay, so first we're going to look at the levels of our metadata right now, just see kind of what things are set at, because we don't know yet. So this is great. It's called what we wanted it to be called, stellar. So I'm going to now name that as our original identity. So orig ident, great. And we're going to add a bit more metadata to this. So here, this is kind of like a 3-step process. I'm going to look at, I do who, what, and then where. So who, so what cells are we wanting to rename right now? We're wanting to rename the ones with this identity, which in this case, in this metadata identity class, happens to be all of them. So we're going to grab these cells. So this is the part that can be changed in here in idents. We're going to call them what we're going to call them. So for what do we want to call them? I'm going to call this one for this new metadata class, which is going to be sample type. I'm going to say that these are all PBMCs. These are all peripheral blood monocytes. So here we go. We're going to go and hit that. And then where? Now, where do we want to stash this new identity? So this new label we've tagged on to the ones that were called this. We're going to stash that into a new identity class called sample type. Great. And what this does is this will now change the identity class to the one we just used. So let's look again at the level of what we're looking at. So this is our pre-filtered object. Now all the cells in here are called just PBMCs. Okay. We're going to do that again, and this time we're going to label it based on the 10x machine that was used, because that's the difference between this data set and the other one that we're going to be integrating with is the machine that was used. So this is our actual variable. So they're common for what sample it is, different for the machine. So we're going to run these 3 steps again. We're going to call it Chromium controller, and then we're going to, oh no, oh yes, okay, this is a normal thing. So then we're going to call it 10x machine, and then you do get a little bit of a warning. It says it doesn't like the syntax. It's okay. You will still see when you do your levels that it is called Chromium controller. So we can ignore that and kind of keep going forward. All right, how are we doing? Is everyone with me still?
So the warning, it says new object name is like 10x machine. So that I can still use the name we put in.
Yeah.
I can still use PBMCs for the rest of the code. I don't have to use X10X. I
would just ignore that warning for now. It's just the syntax for what the label was initially. I believe it makes it with like instead of it being 10x machine, it just added an extra X in front, just so that for the computer, it can interpret it better.
Oh, variables shouldn't start.
They shouldn't start with a number. Yeah. So it'll change it for you.
So like what other kind of errors can you get? So it's a warning, it sounds like they're like, please don't do this, but we'll let you.
It's like an FYI with the warnings. You might find down the line that things might not work, but I don't think I can go over all the types of warnings you can get. There's many, and as you go, this is your first warning. Oh yeah, don't worry. You'll get warnings. It's the errors that will cause something to fail.
Okay.
The warnings
can be ignored. The errors are like,
yeah.
Okay, that's what I was wondering.
I wouldn't say totally ignore it. I would say read it, interpret it, know what it's saying, and then. But it's not directly. Yeah.
And it's not changing anything. Like.
So what it did is it changed the new object. Like basically what it was changing it to was to make it. You can't really see well here, but to be X 10X machine
Oh,
instead
so-
of 10X
Okay,
machine.
you did that. I see. I see.
Yeah,
I thought it was like you can't change it to whatever PMCs or something, so we're just gonna call it.
no, we're down, we're
Okay.
down. So we're on line 70 right now. You should be down there with us, I hope. You'll probably see the error soon if you haven't ran this yet. All right, great. So that's awesome, but we're gonna go back to our original identity class. So you can just run those last two lines there and get our identity level set to PBMCC. Okay, so next thing we're gonna do is add some metadata labels to this, and this is going to be metadata that will be used to help filter and threshold our data. So first thing I want to add is our percent of mitochondrial genes, and after that, our percent ribosomal genes. So remember, our data is now aligned to a reference genome, so it has a bunch of gene names, and so the mitochondrial gene names start with MT, ribosomal gene names start with RP. So I'm basically saying all the genes that have this pattern, we're going to store the percentage of those genes that are expressed into this metadata category over here. The next thing we're going to do is get some metadata added about our cycling cells. So here we go. We're going to get a few more warnings, and it's basically just saying that there are certain genes that aren't expressed in this data set. That's okay. Great. And so now we're going to look and see how the metadata was added to our object. So
this percent MT, is that going
Yes.
to count? That's just a percentage of how many of the features are
mitochondrial genes.
but not what percentage of the counts are mitochondrial.
It'll be looking, so the mitochondrial percentage, we'll see it in a bit, but it'll be looking at per cell. It will look at the counts bit. So per cell, it will calculate how much mitochondrial gene expression there is per cell.
Okay. So it is looking at counts.
Yeah, it's looking at counts. Yeah. Pretty much everything here is going to be looking at a mix of counts and so how many of a certain gene is expressed and then which genes per cell. So let's pop on over to our environment over here. And so our object right now, it's called object prefilt. Let's hit the little spyglass here and open up this object into our lovely UI. And we're going to click metadata here and see what we added. So as you can see with our first warning we got, it has changed the name to X10X machine for this identity class. And then it has one level, so Chromium controller. The ones we just added, percent mitochondrial, like percent MT, percent RB, these values here are counts of how much of your gene expression there is for cumulatively of all the genes that start with MT. So this is a number here, and then same with S score and G2M score, except I believe these two are normalized and centered around 0.
So the ribosomal percentage is tiny
As it kind of should be.
because it's poly A prime.
Yeah, because it's poly A prime. So ribosomal percentage is small. If you have some random cell that's through the roof with a ton of like ribosomal gene expression, potentially filter that out. So we're going to go into that now, how to go through that stuff. So scroll back down. So back to section 2, template metadata. Now we're going to, okay, first we're going to save the object where we are right now. And this is going to be saved into our objects folder, and then the name of the data it came from. And then I have the ending bit basically just saying what this object, what stage of the process is this at. So it's our pre-filtered object. We haven't removed any cells yet. Okay, the next thing we're going to do is start to visualize and actually look at our data. So we're going to do a VlnPlot first, showing the number of different genes. So that's N feature RNA. So that's the different genes or mRNA or cDNA that are basically in our in each cell. The next one is going to be count. So that's just going to be, doesn't matter what gene it is, how much RNA there is. And then we talked about percent mitochondrial genes and percent ribosomal genes. Okay. So this VlnPlot is specific to Seurat. So I'm gonna just zoom out a bit so people can actually see the plot. So here we go. So now we're seeing here just a VlnPlot, which essentially is a fancy version of a box plot. And we see some cells on it as well. So each dot here represents one single cell. Lots of dots represent lots of cells. So for a cell up here for end feature RNA, it has under 8,000 features. So under 8,000 different types of genes in that cell. That seems to be a lot considering the majority is down here. So depending on your cell type, you might actually want to filter out and set a cutoff point for the number of genes you have, number of different genes in a cell being expressed. The reason for that is because there could be multiplets or doublets as part of the droplet-based library preparation process. So when you have your cell that goes and connects with, you have your cell in your bubble with the gel bead, usually it's one cell, but if you have stickier cells like macrophages, like me, you might get a few cells in there. And that's going to actually really skew the data that you're looking at down the line for differential gene expression and how many genes are being expressed in these certain cell types. So those outliers due to that, those errors should be mostly filtered out by this point, but we don't get them all sometimes. And so if you're seeing some outliers like that, you might want to set some maximum thresholds. Okay. So another thing we can do to look at is the feature scatter plots here. And really quick, I'm just going to save this before I forget. Great. And so these are all going to be saving into your folder under pre-filtered outs. Okay, so next you have your feature scatter plot. This is another way to visualize any outliers. Now, this is where things get a little crafty and very subjective, honestly. So Seurat kind of recommends to not use statistics to get rid of a certain percentage of cells way out here or a certain percentage of cells way down there. Instead, they suggest looking at it, seeing where the pattern kind of falls off a bit, and then cutting a threshold there. So that's one way to do it. There are plenty of scripts, and basically scripts in R available to cut off things based on statistics instead, if you want to do that. If you're setting up an automated pipeline and don't want to look at these plots every single time, that might actually be the more efficient route to do. Actually, it would be the more efficient way to do this, but it's not recommended by Seurat. So there is this kind of back and forth debate of how we set these cutoffs off, how we threshold our data. One of my supervisors kind of said, like, do we even need to get rid of anything? And the answer is, it depends. Sometimes yes, sometimes no. So this process of filtering that we're going to do should be sample specific every single time. Every single time. You should not integrate all your data together and then try to filter. This should all be based on the individual sample, these filterings. Okay, so that's another visualization plot that we can look at for quality control. The last one, we're going to look at our VlnPlot, just showing our cycling cells. So these are cells in the cell cycle. We have a couple, maybe. These are still pretty low scores here, but we will kind of cut off a few of these, I think, with our thresholding. So now that you have these quality control plots made and generated, you can take them into consideration when you set these threshold cutoffs. So for this, I'm going to get ready to threshold. We're going to threshold down here. I'm just going to first rename, change this object. We're going to go from this pre-filtered object. We're now going to call it filtered because that's what I'm about to do. And so if I filter it, look at what it looks like, and I don't like it, I can always go back to my pre-filtered object. Plus, we already saved it initially as well. This is just a quick way to go back to that. So based on what we saw, I'm going to set this cutoff threshold for the number of different genes per cell, which is going to be our end feature. We're going to set that at 7,000. The next thing we're going to do is we're going to make sure that the cells that are in our data set have less than 10% of mitochondrial percentage. I've seen this be anywhere from 5 to 20 that you could use for a percentage of mitochondrial genes. And the reason we do filter out those that have more than 10% or more than 20% is because we start to see mitochondrial leakage when cells get stressed and are like dying. So we don't necessarily want dying cells in our data set if we're trying to look at what a healthy typical cell from this sample we have would be like. So we want to get rid of some that might have been very stressed or dying during the library preparation process. So during that chip process of 10x, that very first day. All right. The other thing too, I'm just going to set we're just going to make sure that no one's cycling, no one's in the cell cycle for these cells. If you're looking at cells or looking for cells that are in the cell cycle, don't do this. Or if you're looking at, yeah, that would be the biggest thing I'd say. If you're looking for cells in the cell cycle, don't filter them out, obviously. So again, all this stuff is laid out for you, but take into consideration what you're looking for, what you're looking at, and why. So
those filters are just based on expression of certain genes.
These filters were based on the plots we were looking at. Yeah. So if we go and look at the pre-filtered one here, for this, you can see that there are just like a few cells that are in the cell cycle potentially. And the cell categorizing those cells as being in the cell cycle is based on a large list of genes, I would say, a large list of genes that are upregulated when cells are cycling. And so now if we go look at the plots, we're going to look at our filtered object. We're going to see that this is what we have left. So our cutoff was at 0.5 for both of them. And we have a nice evenly distributed VlnPlot. Going back to the other ones as well. Here's our VlnPlot for percent mitochondrial genes. I was pretty aggressive with cutting it at 10. You can do 20. I think 20 here is completely fine. Even 25, maybe 30. So this is the subjective parts of thresholding data for single cell RNA-seq is basically what cells you want to keep in, what cells you want to keep out. Okay, we're going to save that one. And then the other feature scatter plot we're going to do, it's going to be this one. It's going to save that and then I'll pull it up one more time. Okay, so now we see here, so we set the cutoff for number of features, so number of different genes per cell. We set that cutoff from 7,000, and we have an increased correlation now between our amount of RNA per cell to the number of different genes expressed per cell. So we like to see that high. And then we talked about this already. So these are our cycling cells. Okay, so I'm just going to save those. And now that we've filtered our Seurat object, we're going to save that as our filtered Seurat object. Cool. And that took a little bit longer. Okay. Any questions from anyone in person or in the chat on that? I don't see anything in the chat. You guys kept up easily? Yeah. Okay, great. Yeah, it's all written out, so it should be.
You did a great job.
Oh, thanks. Yeah, so the code for everything should be pretty clear by this point. This is now where it gets very streamlined. Okay. Does anyone want to comment or discuss different filtering processes or things that they've seen at all? Because it really is kind of a big debate. We're all pretty new. Okay. So yeah, the biggest thing I want to just like reiterate over and over again is your data isn't going to be as clean as this. You're going to have to make some judgment calls with whether to keep cells in or kick them out. Totally up to you. You can always go back to these stages at different points and make different versions of your data set depending on what your outputs are and what you're seeing. This is not an over manipulative thing to do either. This is very just clear cut what cell you keep and what cell you get rid of. We're looking at the raw read counts here. So, okay. Okay, so We're going to move on to template number 3. This one's even easier. So this is going to be for removing, we're going to get rid of that donor name. We're going to be removing any cells here that are potential multiplets or doublets. So I'm going to actually go to the slides and just get the slides caught up with everyone first. So we've gone through SoupX. The next thing we just did was we imported those outputs from SoupX to Seurat and we labeled them. We just added some metadata labels. Next thing we did that quality control. So we looked at our raw data, made some plots of it. So we have a sense of what the gene expression is like in different cells. And then we did a little bit of thresholding. So we got rid of a couple cells. The next thing we're going to do is remove any artificial doublets using a package called Doublet Finder. So if you go to Doublet Finder's GitHub, they also reference at the bottom of their main page of their README file, they reference a bunch of other Doublet Finder packages. And I, again, encourage you guys to see what works for you. Doublet Finder worked well for us. I like the way things visualize, and it's also widely used. So for detecting doublets, this is, again, where you end up, instead of having either no cell per gem or one cell, as you should in a gem, sometimes you get more than one cells. And so this is the part that doesn't get filtered out as much from the cell ranger count function from that pipeline. They'll get rid of cells they'll get rid of things that might be cells that aren't cells because they don't have much mRNA expression, whereas they won't get rid of ones that might be 2 or 3 cells that were labeled as 1. And if you have 2 or 3 cells labeled as 1, by the time you're in RStudio, you have no idea. And you can be running downstream analyses on things that are incorrect and not biologically accurate. So these usually stand out when we're thresholding and we're getting rid of cells with way more like number of features, different features, or different quantities of mRNA expression. We can usually cut them out, but this package is specifically designed to detect doublets. So we use this after we've done our normal thresholding process. All right. Okay, so open Doublet Finder template. Once again, we have our names. We're going to now create directories for our Doublet Finder outputs. Great, that just appeared, and we're going to name that path. I'm going to run that as well. Okay, so we were using the name object filtered. We're now going to call it pre-Doublet Finder because that's what's happening with my naming. I like to be very organized and very clear of what's going on, so I know what's happening with that object. It's really easy to accumulate lots of objects as well. So we're gonna, if you don't have this already, which you should if you're all following along, this is just the path to go get the object we made. Okay, so before we get into this, I just want to show you. So with Doublet Finder, this is a package. It also has a bunch of step-by-step things. I've turned it all, like the whole process, basically into one function with some variables here. Okay, so the number one variable, or the few variables you'll want to kind of adjust are going to be your dimensions, your PCs, so your principal components, which I usually leave set at 1 to 15. But if you can also go 1 to 30 for this. I would say be generous with the Doublet Finder part because this isn't your fully processed Seurat object yet. But for the data that we're using today and for data I've used in the past, I find 1 to 15 be pretty good. This variable here is going to be your expected doublet formation rate. And this comes from a table from 10x. And depending on how many cells you loaded onto your chip, your Chromium chip, that's going to determine how many multiplets or doublets are expected just based on your numbers. So this can be looked up if you're doing your single cell RNA sequencing library prep with 10x, it'll have the table there, and you can basically just run your finger over and you're like, all right, for like 2,000 cells, or I don't think it's 2, for like 5,000-ish cells, it's about 8%. For these cells, we loaded on 500. We expected 500 cells. We loaded on 1,600, but we're expecting 500. So for the number of cells we had loaded, we would want to put a variable in that accurately represents that. So I actually put this in based on 500. So that's 0.35% or 3.5%. Okay, so that is one variable that you will change based on your data. So this function will go through, it'll do the typical processing steps, which we're going to get into a bit more in the last one. And it does a lot of quality control. It basically figures out which variable parameter will work best as part of this function, and then this as part of its quality control. And then once it has that variable, this function takes that variable, brings it forward to then run doublet finder on it. So again, there's a lot of resources available. You can read up on this, you can read up on others as well, and you can go through all the code with this too, but this is just a simplified, easy, streamlined way to go through and run doublet finder on your data. The other thing that's different is we're using ST transform compared to scale normalize and scale normalize center with Seurat. Okay, so step one, even though it's at the bottom, is you're going to want to make sure this function exists. So you're going to run this. And
I ran the function.
yeah, you're not going to be able to run the stuff leading up to the function because you don't have the function loaded. Yep.
Thank you.
Yeah, so even though it's at the bottom, load that, and then I have it at the bottom just to kind of keep it out of the way.
Okay.
So once that's loaded and you have the function, you're going to run doublet finder on that object. And so these arguments here, even though these are our default arguments, you can adjust them when you run the function itself. So as you can see here, I've changed our resolution from 0.1 to 0.5, and I've also tweaked this X variable. It's the X variable that is specific to doublet finder. And then you're going to hit run. Oh no. Oh no. This one? No, I think it's
Did load function and then to write before load function.
I'm just going to see what's up here. Just going to comment that out. And try again. Wow, I really hyped that up and then fall short. Okay. Oh, did I actually not run that?
Yeah.
Okay, thanks, Phil. Okay, it should run. And then feel free to correct any of my concussed brain typos that you see in these scripts. There's a big disclaimer with that. So this is actually going to take a little bit of time to run. The larger the data set, the longer it's going to take, just so you know. So we're going to let that go. And while that's running, we have a little workshop task for you. And so I'm going to give you maybe like 15 minutes to do this. So we'll kind of check back at 1:40. But our SC process task here with our markdown is using what you learned today with these template scripts. We'll have you start with script number 1. Go back through and prime it so you can do everything that we did with PBMC-C with PBMC-X. So that's our workshop kind of test for you. This is actually, I think, almost done. So we might actually be okay. Yeah, so that'll be the task to do your take-home assignment if you want. And again, it's going to be really simple. You're just changing the names of things and getting that to the point that it can be integrated. Cool, so that was super fast. Okay, so now that that's done, go ahead and use these 3 examples here, 1 through 3. And do the same thing for PBMC-X, okay? And I'm going to do the same. And if you have finished running your doublet finder, just make sure to save it. Okay, I'm going to stop share for now and we'll circle back at 1:40. Just going to pause actually.
Function.
I moved it. I had it at the top, but I wanted to talk about the steps and then I ended up not moving it. I moved it down and then I was like, at the bottom sheet.
It's an old, yeah, it was Perl coding convention. So if you ever coded
Hey, how's everyone doing? You still have 1 minute, but give me a thumbs up if you're already done. All right, Daniel's done. Everyone remembered to unzip or basically decompress the matrix files as well. Okay. You're welcome. See ya. All right, so we've ran Doublet Finder. You should have gone through and done the same thing for PBMCX data. If you have any questions on that or something didn't work, please comment, please put it in the chat. Otherwise, I really want to see a thumbs up to make sure that we're all on the same page before we move on to the last step, because having both of those is needed for this last step. Any more thumbs up? Friends, okay, we got 2 total. All right, I'll let you guys get those thumbs up while I'm kind of just going through, and we're just gonna talk about the next step. We're just gonna integrate these 2 data sets, which when you have multiple samples, you should be processing everything that we did up until this point should be on a per sample basis. And if you're doing a well-controlled balanced experiment, you should have multiple samples. I do expect with these experiments that you're going to want to integrate these multiple data sets together, get them all into one Seurat object, and then you can easily compare between the different samples and see what's going on. So we're going to get this fully processed Seurat object of our PBMC samples ready and see what we can do with them. All right. So everyone should see RStudio now again. The last script we're going to open is going to be template number 4. And this one I just titled processing. So we're going to hit play. We're going to get our libraries loaded. The next thing we're going to do, I'm not on the right one. All right, so we're going to get our libraries loaded with Seurat, SC-transform, and ggplot too. SC-transform, it's important to note that SC-transform is different than if you go through Seurat and follow their online vignettes. You're going to go through a different step of processes here, whereas SC-transform actually kind of replaces the normalization, scale, and transform data functions. So on Seurat, they do talk about using SC-transform, and there is a vignette for how to use SC-transform on Seurat objects, and it works a little bit better. And there is an SC-transform too as well. So this is the part where you're scaling and normalizing, centering your data. So it is an important step. So I do suggest using SC-transform over the typical Seurat process. This is like the one time I'll actually advocate for something. But of course, there's other ways to do this as well. And so if there's something that works better, go for it. I like SC-transform and I'm the advocate for it. So we're going to use it. All right, so we're going to prep our objects for integration. So we have our 2 objects here. For every object you have, you can just make another version of these 4 lines. So we've got name number 1. I'm basically just going to be, so this is just to load the objects. So name 1, you're going to load in the object and then, and read it in if you need to. But for now, we have our 2 objects already made. So we've got PBMCX and PBMCC coming from the objects we just had. So we're just going to run that last line here of the ones that we made. So in our data under Doublet Finder, under objects in each of our data sections, we should have the most recent one should be Doublet Finder here. So this is the one I'm going to load right now. And, oh, not open connection. That's weird. Okay. Got to run the first 2 above, and then we got to run that. Okay. And so now let's look at our object that we just loaded. Okay, so we got a summary of our data here. So we've got an object of class Seurat. We have almost 30,000 different types of genes across just under 500 samples. So we've gone from, we expected around 500, so we're kind of right on the money here. And it's within 2 assays. So we have 1 assay, which is our SCT assay, which is from back when we ran Doublet Finder on it. We're going to replace that. And And then we have our other assay, which is RNA. And that is our raw, this is our assay that we haven't manipulated at all. We also have two-dimensional reductions. And this is all because we had to run some of this processing stuff in order to properly run Doublet Finder. We're going to replace all of that though now that we've removed some of the doublets. Okay, we're going to load the other one we just made as well. And let's take a look at PBMC's X. There we go. So this is super similar, basically exact to what we had up here. They are different samples. They are just super similar because they might even be from the same sample or same donor and same things loaded, but they are different cells, and we're going to integrate these two data sets together. Okay, so we're going to create in our SC process folder. So I'm going to pop back to that. In our SC process folder, we're going to make a new folder now called integrated. So these folders house the data that was separate. This one's going to hold our combined data. We're going to name that file path, and we're going to give our object a name as well. Okay, this part, if you have a lot of objects to merge together, this part can actually be really slow, and that's based on the size of the gene expression matrices. So if we're merging multiple gene expression matrices together, this is the function we use. This is not integrating, this is merging the matrices together. So then the more you have, so we've got one here, the second one's here. If you have more, you can just do C, open brackets, and then list them all out. And as long as you do that, you then just need to update what you call them in the same order as they are here over here. So this is how you assign the names. The add cell IDs argument adds the names and names these different objects. So we've already combined those objects in objects combined. Next thing we're going to do is we're going to run this process all the way down. So we're just going to hit play and get started. And you're going to watch it go up in the console. So first thing it does, and I don't know why this works, but if you want to run, if you want to integrate them, but also run SC transform function on each individual data set first. And all pairs, that's new. I'm going to run that step at a time, one step at a time. So it should split them and then run SC transform on each of them individually. But in order to run SC transform on them all individually, you have to kind of merge the matrices together first, and then you split it. This is a weirdly critical step, and then run SC transform. The next thing you want to do is select integration feature. So these are going to be the genes that the integration function is going to base things off of for figuring out what cells to cluster together down the line. Okay, we're going to try this one more time because I'm not actually sure. Argument to which object classes. So I haven't had this error before, which is odd. Int list, is it correct? Use names true. Okay, so I haven't had this one before. So I don't think I can troubleshoot this live in front of you guys. That one's not going to work. No. Okay, so we've got prep integration in list. I'm not actually sure what's going on here. Okay, I'm still going to walk you guys through this, and then at the very least, the code will be updated and put on the GitHub in the same folder that you copied this all over from. But I do want to walk you through this because we only have 12 minutes left still. So the next part is it's going to find the integration anchors, and this seems to be something to do with this function specifically. So I'm not sure if something updated recently because this did work yesterday. So I'll check on that, but find the integration anchors, and then it integrates these data sets based on similar features that are highly expressed or lowly expressed in cells. And that's how it's able to find and map out which cells are similar to each other to integrate together and put together.
Try int underscore anchors.
Int underscore. Really?
Yeah.
I don't think it's going to be a naming thing. I think it's actually looking at the error. I think it's finding all pair. I think this is going to be like, I'd have to go in and see what's going on with this source code, because this is not for the which. That's kind of a deep thing for object classes. They should both be. Not that loaded, not that loaded.
Or when you
what?
did that, you just added a new. Is someone in the chat?
Yeah, let's see what's up in the chat. Code. Oh, good. Okay. It's working for
You
someone.
probably missed a block.
I think I either missed a block or I don't think I missed a block, but we'll run it this. Okay, ran good for you guys. We know that exists. That's the fine warning. Okay, I'm gonna hit run again. David, you're giving me, no, Daniel's giving me hope. Okay. Can I get thumbs up that it worked for others? Might just be a me problem. Yep, good. I gotta trust my code. Okay, so I'm having a weird issue here. I can pull up a different RStudio environment, but it looks like it's working for everyone else. So that's good. So it might be because I reinstalled or updated something, but after you have that, it sets your default to say to integrated, and then it runs these pre-processing steps. These are pretty standard. This is gonna generate, it's gonna do its principal components, then also run UMAP. You can also add in the run tsneap function if you want, although UMAP seems to be a little bit more preferred in this field and seems to be a bit more, I'm a fan of it. And then you can plot the UMAP, showing basically your data itself. So what I'm gonna do is I'll just run this without merging the objects together. So let's see if maybe that's an issue. Okay. I guess I can't really integrate it, but what I'll do is okay. Don't do what I'm doing. I'm just going to get so I can show you similar plots and we can keep moving forward with the visualization. Oh, we don't have that. That's fine. Okay. So now we can pull up a UMAP plot of what our integrated object looks like, and we'll pretend that mine's integrated. Woohoo! Okay, so as you can see, there's not a ton of cells. I vaguely know what yours looks like. I have seen it before. I could pull it up, but we're just going to keep going here. So you get to see your UMAP object, and the big things here are these clusters. Everyone's obsessed with these different clusters of different cell types. So cells are clustered based on their similar gene expression profiles. And then the closer two dots, and a dot represents a cell in this UMAP projection, the closer two dots are together, like these two here, the more similar those two cells are. And so based on that, these cells also get colored and put into different clusters categorically that are going to be showing how similar, like this, apparently between this big guy here and this little one here, these are technically the same cluster. However, on the UMAP, they are a little bit of a distance apart. So I would kind of question whether that actually is the same cell, but for you guys, you probably have this one showing up potentially as its own color, cell colored cluster, which would be more accurate. So to tweak how many clusters you have, and this is again going to be based on the types of cells you've loaded and how many different types of cells you know are in your sample. To tweak the different numbers of cells, you're going to adjust this parameter here. It's your resolution. So let's say I go to 0.1, make it super small. This is going to give us fewer different cell types or fewer clusters. Okay. For this part here, I like to say that it's better to overcluster than undercluster, because if you're underclustering, you might be missing some sort of cell type that you wouldn't know exists because it's buried in with other cell types. So I moved it back. It's better to overcluster than undercluster because you can always group different cell types together. It does make cell labeling a little bit harder though, because you're going to have all these different clusters to label and you might not know what cell type one of them is. So let's make this super big. That's not super big, but we're going to make it like that. Great. Okay, so this now, they've all became like this, and this group over here has split into 3 different clusters. Okay, so once we have the UMAP, the other thing I can't do, but you can do, is to run the UMAP and split it by original identity. So this is going to split our 2 samples up, and now you can see how the UMAPs of those 2 different samples differ. Great. Okay, so when you're doing just processing and labeling of metadata, you can use the integrated assay, which you're in right now. I'm not. But if you're ever doing downstream analysis, you never want to use the integrated assay. You want to use SCT, which is going to be your transformed data or scaled and normalized data, or your RNA data, because these are going to be more accurate. Integrated assay is only going to take a subset of your features. Here you go. You can see how many you took in there to merge things together. And then if you do any differential gene expression on that, it's only going to be looking at those features in the integrated assay that you indicate there. You can always increase it. It'll just take longer for this function to run and might take more time. Other things you can do, you can find all the markers. So this function is basically going to pull all the different types of genes that seem to be good representation, like genes that stand out in a certain cell, certain cluster of cells. So these are going to be marker genes. And you can use these marker genes then to label the data set. So I went through, found marker genes for all these different types for all these clusters. I'm going to organize them with this function by log fold change and just make a CSV of that, and that will save as well. So these are being saved right now into our integrated folder, and then I just saved cluster markers there. Cool, so CSV files are great. Let's see what a heat map looks like. So we just did that again with cluster markers. We can do our do heat map function, and voila, you have a heat map here. You're gonna have to open it up. Make sure you can see it, make it really big. You can always reduce the number of genes you have or change the dimensions you see. But for cluster 0, you have these genes up here that are lit up in yellow along with this batch and this batch. Kind of seems to me that cluster 0 might have a couple different things going on. It might be a mixture of, might be very similar to, looks like cluster number 3. So when you guys do this, I do believe you guys will have a pretty clear cut light up section of yellow for 0, then 1, then 2. And it should be very clear and obvious in your data that you're looking at what markers are with what cluster. And then you can look at what those genes mean. If you don't know, which I'm Googling things all the time with these genes, you can look at these genes and see, okay, of these genes, what cell type do I think this is? So looking at this, I see a lot of HLAs, I see LYZ. I'm thinking this is either macrophages or monocytes at this point. So that's another way that you can generate a plot and a tool to label the cell types. And then lastly, you can look at the different levels you have, and this is going to pop up and show the numbers that represent the cell types. So if you want to label your cells, let's say you went through and you know what cluster, like cluster 0 is this type, cluster 4 is this type. Once you know that, you can label your cell type by just listing them all out in the same or the names that you want to call them. So list out the names you want to call them in the same order that you have for these values. And that'll stash the identities. It'll save the names. This won't work because I have more than 8 clusters, more than 8 cell type names, but I have only 8 clusters. So I'm not going to run this, but you can, and you can label all the cells. These might not actually be what cells you have. These might not be the cells you have, is what I'm trying to say, because things are going to be different for your data set, so, and how things clustered, but it is likely. So you can go through and check and see which cells you have using these different plots. Last thing you can do is make a dimensional plot here in UMAP reduction. Yeah, UMAP reduction, and then have the names of your cell types labeled on the UMAP plot. And now you have your fully processed Seurat object with your integrated data that can be used for all the fun downstream applications. Okay. Thanks. Pardon?
Is that it?
I have a question, or someone said something in the chat.
No,
Have to run for another meeting. Okay, perfect. Yeah. Okay, you guys do have me for maybe like 5 minutes. If you have any questions, it would make me feel good to see that, like a show of thumbs up, knowing that the code ran for everyone. Awesome, Daniel gave a thumbs up. Okay, if it works for Daniel, it probably worked for everyone else. It worked for everyone. That's all I need. Thank you, Franz. Okay, great. So we'll just finish up with our slides. Key takeaways. So use these scripts as a tool to help with your single cell RNA-seq processing. Keep them, they're yours. Use them, use them, use them. It's a resource for you. There's many packages out there and many ways to pre-process data. That's a take-home message. You'll need to find what works best for you and your data empirically, okay? Here are the list of the resources I use today. So definitely if you're using these packages, read up on them, know them. If you're doing a thesis, you're probably gonna get asked about them. So know them. And then I just want to acknowledge my lab, the Lynn Lab, and the Precision Health Initiative for also giving me funding so I can also do all of this stuff. And that's everything I have for today. All right, thank you, everyone.